In this meeting, I'm presenting results from a massive single-cell RNA sequencing study that we've put together of tumor and immune cells from patients with MGUS, smoldering myeloma, as well as overt myeloma. We're talking 877 samples, 533 bone marrow and peripheral blood specimens, and 365 individuals. 241 of those individuals have both tumor cells and immune cells sequenced at the same time to enable integrative analysis of tumor and immune cell biology. Overall, we're looking at 6 million cells, which is, a truly fascinating data set to be able to work with. And the goal here is to identify immune biomarkers of progression, but also understand how tumor cells and immune cells are co-regulated within the bone marrow microenvironment, the hope being that we can then disrupt these interactions and develop new therapies. And so we have been able to discover a number of new immune biomarkers. We see a lot more cytotoxic T cells in patients with disease progression, and this is independent of chronological age. We also see more clonal T cell expansion with disease progression. We see changes in the cytokine profile of myeloid cells. They obtain a more pro-inflammatory and pro-tumorigenic profile with disease progression. And we see increased interference signaling. And what we find is that interference signaling is active both broadly across all immune cell populations in the bone marrow, as well as tumor cells. And leveraging that tumor cell correlate, we've been able to validate these results in external cohorts and hopefully help bring those biomarkers back to patients.